Hello, Mr. Allman here, and this is your presentation on biotechnology. I'm hoping that through today you can get a little taste of each of the different techniques that are necessary for you to know in order to understand what biotechnology is about, and some examples of some of the applications in which how these different techniques are going to be used. The first one is restriction enzymes. Restriction enzymes are particularly important in terms of a starting point, a way to be able to chop up DNA, because the genome, even of very simple organisms like E. coli bacteria that are used a lot in research, still have, there's too much DNA to be able to do anything with, practically speaking. And so it needs to really be divided up into smaller bits for a variety of purposes that we'll get into. So restriction enzymes, they have official name of restriction endonucleases. A nuclease is an enzyme that will cut DNA. And so each enzyme is going to cut a DNA sequence at a particular site. So there needs to be a 6 to 8 base sequence, which the shape of that will match up with the active site of the nuclease. And then at that point, it'll act like a scissor, and it's going to cut that DNA at that point. Hundreds of these have been isolated, so these are naturally occurring things that most likely existed as a natural defense, that bacteria are producing these restriction enzymes because the disease that impacts bacteria are a type of virus called phages. Now a phage will just like any other virus, it will inject its viral DNA into the bacteria to infect it and try to take over. And so the bacteria would have these particular restriction enzymes that would be able to recognize that viral DNA sequences and then cut the viral DNA so that it can't carry out its goal of trying to cause the bacteria to make more viruses. So for that reason, that's why there's a variety of different types. Different types of bacteria have yielded different types of restriction enzymes, and each of those we can then use as a way to be able to cut DNA at different points. So EcoRI is one of the most famous ones. It comes from E. coli, so these things are usually named after the bacteria that they come from. And the particular sequence GAATTC is the place at which the DNA, so you can see the protein is here on the right, and then the DNA is cut at that point, so the enzyme works at that spot. Each enzyme is going to recognize a different site, so whatever that particular sequence is, it'll cut it there. And again, we could have a difference between those that have the sticky ends, so where there's an overhang of DNA, so it's not cutting it clean through, but it's cutting it at 2 different points on the 3' and 5' ends, or those where they have a blunt end where it does cut straight through. But these sticky end ones are the ones that can be useful for us to be able to make recombinant DNA. So ligase is the other enzyme that you need to know. So after this part is cut, in order to get the DNA, whether it's adding in a new fragment or just to connect the pieces back together again, we need another enzyme, ligase, to be able to stitch together the sugar phosphate side chain that's holding the DNA structure together. And so that's how we can make recombinant DNA. Interesting addition is that often restriction enzymes work on palindromic sequences. So palindromic sequence meaning that if you read it backwards, you're going to get the same sequence. So GAATTC for EcoRI, when we look at the complementary base pair, if I read from right to left, I get GAATTC. And so it seems that viral DNA was likely had a lot of these palindromic sequences in them, and that's what the bacteria produced enzymes to be able to recognize. Next factor here is vectors. So vectors is one of the techniques, something that's going to be made using restriction enzymes as one part of that. So a vector is going to be a DNA molecule that can be modified so that it can be injected into a target organism, and then it will express its DNA. So there's a lot of types of these. The one that we'll cover in class is bacterial plasmid, but other things, phages, viruses, and artificial chromosomes can all accomplish the same thing. Now, the idea is that we need to be able to take a piece of DNA, like a plasmid, and we need some way to to do two things. We need to be able to make sure that the host, in this case the bacteria, will make the DNA that's in the plasmid, and we need to make sure that we can recognize that it's actually making that protein. So that's why we need a couple of things. First, we need an origin of replication. So this ORI, so this means this is a sequence of genes that will have all of the necessary coding regions to ensure that transcription begins, so that whatever genes are located in the plasmid is actually going to get made into proteins. So we need some spot for things like the RNA polymerase and transcription factors to be able to bind to. We have a region where we're going to have the genes of interest, so whatever gene it is we want this particular plasmid to contain, that's going to get inserted here in this next section. And then we're going to have some type of screening, some way to be able to recognize them. So in this case, ampicillin resistance can be one. So if you see here on the left, we have a sequence of genes that will give the bacteria cell ampicillin resistance, so that if we take some bacteria that are not resistant, we insert this plasmid, only those that are able to grow in the presence of ampicillin will thus have the plasmid, and so we know that those are the bacteria that will be capable of producing our genes of interest. So there's a size limit on plasmids, so because those are small circular pieces of DNA, we can only put so many chromosomes in any, sorry, not chromosomes, we can only put so many genes in any one given plasmid. So that's led to the idea of making artificial chromosomes. So if we want to work not in a bacteria, but if we want to work in something larger, say, for example, yeast, we can create chromosomes that would have potentially hundreds or thousands of genes on them if there's a greater number of products that we want to make. Right, so if we go over to some applications. So the main application of this is genetic engineering, and bacterial transformation is the primary example that you should be aware of in terms of how genetic engineering works. But this is something that's happening in all different types of organisms, not just bacteria, but in, as I said before, yeast and C. elegans and in fruit flies and a number of other mice, any type of organism. We have the capability to do this, but there are just certain organisms that, for the sake of convenience and familiarity and the ease of working with, we've chosen certain species, and E. coli is one of those. So if we take DNA from a different organism and put it into an organism of different species, we're creating what's called something that's transgenic. So transgenic means that it's spanning across, it has DNA spanning across multiple species. And so we need a restriction enzyme to be able to cut DNA, and we need to make a vector, and so those are the two things I just described. So in a bacterial transformation, what's happening, we take the bacteria, we take a bacteria that could be from a plasmid from that bacteria or a plasmid that came from some other source. We are going to use a restriction enzyme to cut that plasmid open. We will insert in a gene of interest, so a piece of foreign DNA that could be from humans or from another E. coli or from some other organism entirely. It's important that plasmid also has a marker in it, something like the GFP protein or ampicillin resistance. So we create that plasmid gets inserted into the cell, and so now that bacteria cell will express that particular, the protein, the genes that are on that particular plasmid. So it will replicate and will make copies and copies, and a few examples of some ways in which that can be helpful. So we want to do research. So we want to do gene research. We want to make copies of particular genes and look for development of things like pest resistance. So if we want, if we can find in one plant a gene that will make a plant, a crop resistant to pests like mayflies or aphids or things like that, if we can insert that gene into a plant like soybeans, then now our soybeans will have a natural resistance and we won't have to spray additional pesticides on them. That again is providing us with a great advantage. We can insert genes that will provide enzymes for bacteria to be able to clean up toxic waste. So if there's an oil spill, we can use these transgenic bacteria, these engineered bacteria, to spread them onto the oil slick and be able to organically digest all of that waste. Again, it will suit our purposes because it's solving one of our problems. If we have a heart attack, if we have a blockage in one of the coronary arteries, can we make proteins using the bacteria that will allow a less invasive way to be able to clear out that clog so that there's not going to be heart attacks in the future? Someone who is short or is lacking on human growth hormone. If there's a growth hormone shortage, and the person needs injections of that particular hormone, again, we can insert the growth hormone gene into bacteria, have bacteria make that protein, and then provide that protein for the person in the form of injections. So those are some examples of some applications, how we would want to use genetic engineering, how we would want to combine together DNA from different organisms to solve some type of problem. So the screening process, so I've talked a little bit about this so far. The two important ones here are ampicillin resistance. So by growing the bacteria in an ampicillin-rich environment, the ampicillin will kill all of those bacteria that didn't recombine and take the plasmid, and so only those that do survive have the plasmid and thus have the gene of interest. GFP is another important one. That stands for the green fluorescent protein. And so as you can see here, the one on the bottom right, so this one has each of those dots is a colony of bacteria. So that is the growth of many bacteria from one original bacteria that had taken the plasmid and now is expressing a protein that when exposed to UV light will glow green. This comes from a type of jellyfish, and it's abbreviated GFP for green fluorescent protein. The other applications for this beyond genetic engineering include just being able to tag and to target particular genes to see when and where those things are going to be expressed. So in this case, this is showing the larva of a Drosophila. So you can see that the head is on the left, the tail would be on the right, and you can see there's a number of different proteins that have been tagged with fluorescent proteins. And so each of those reporter genes has been inserted into the DNA of the Drosophila, and so when those particular proteins are expressed, they glow with these different colors. So by giving different reporter genes to different parts of the DNA, we can see where it is that different genes are being expressed during different points in the development of the fly. And that's super useful in terms of understanding how it is that development works. How we can go from a single-celled one set of DNA to having this complex array of different body systems and different cell types all with the same DNA. And that all has to do with what proteins are expressed at what point. And it's very difficult to be able to tell that just by looking at cells and looking at chromosomes, but by using these fluorescent proteins we can tell when it is that each of these things are being expressed, and the process of development in a Drosophila fly, even though it seems like an organism that's very different from us, there's actually a lot of proteins that we have in common in ourselves. So this process is really telling us also quite a bit about the development of humans and how it is that a human embryo develops during the early stages. Now, a related process is gene therapy. Now, this is different because this is looking at engineering cells in a multicellular organism where we're actually trying to change the DNA, not in a single cell like in a bacteria, but changing the DNA in many cells in an existing multicellular organism. So if we look at a human, in this case, if we say a person who has, for example, leukemia. So leukemia is a blood cancer, and so that there's the bone marrow, which are the cells within the center of the bone that are making the blood cells. Those cells are cancerous, and so those might need to be removed, or the person undergoes radiation or surgery in order to take those things away. Now, one solution is to do, the current most common solution is to do a bone marrow transplant, where we take the cells from the bone marrow of a donor and give it to the give it to the person who has the cancer. It's difficult to find a match for that, and there are issues of rejection and of the transplant not being successful. So instead, what is always better is if we can put a person's own healthy cells back into their body so that there won't be any issue of the immune system being able to reject that. So in this case, if I have a defective gene in a particular cell that is causing that cancer, I can take a cloned gene. So this is again maybe a synthesized or artificially created or a foreign piece of DNA from another person, and I can insert an RNA version of that into a retrovirus. The retrovirus will inject that viral RNA will convert it into DNA, and then we'll insert that DNA into the cells, into the human cells. Those human cells can then be inserted directly back into the place where they're supposed to grow in the body. And if the virus has done its job of inserting that particular gene into the human cell, now the human cell will have that new unmutated copy of the gene, and it should grow normally. Now, obviously, this is quite tricky in terms of being able to get to the point where we can get many cells in the body all to take up a different copy of DNA, and so that's why this is something that's still very much in the experimental stage. Some other examples of genetic engineering. One of these is the idea of a farm animal. So in this case, we are inserting genes into animals to get the animals to be able to produce something that we want. So if, for example, someone who has hemophilia and doesn't have the protein for blood clotting, it would be beneficial to get that particular protein into people in an easy way. And so one thing that we can do is then to insert that gene into the goat. The goat will produce milk. That milk will have that blood clotting protein in it because we have inserted the gene to make that protein into the mammary glands of the goat. So in that case, we call that a farm animal. But a more common one and a more widely applicable one is that of genetically modified organisms. And so being able to genetically modify foods to be more nutritious would be of great benefit for all people, but certainly those people who are suffering from malnutrition. So the example of golden rice here on the left, this is a rice where the gene to create beta-carotene, which is a precursor for vitamin A, has been inserted into the rice plant. And so when the plant produces the rice, it has additional beta-carotene in it, and so that can be a easy supplement for the diet of the billions of people around the world who eat rice as a staple of their diet. They're now going to get that additional vitamin that in many cases might be the difference from them being malnourished versus having all the nutrients that their body needs.